Okay, so this is about where we left off, and wow, Linda, you look like you're ready to do surgery. That is awesome. No,
Oh, yeah.
keep it on. This is getting serious.
I just laughed bad.
So,
I think she's like a nurse.
just a very quick announcement. We will have a quiz posted tomorrow, and it's going to be over whatever content we covered yesterday, today, and then chapter 3, I believe it is, that we were on in Elephants and Acid. So look for that, and there will be advice in the module. So we left off talking about cells. And what chapter 4 is, it's just a tour of the cell. And so we're going to start going through the structure and function of different parts of the cell. And I'm going to want you to know, bare minimum, that, what the structure and what the function is of these different parts of the cell that we're going to talk about. Sorry, I know that's a little fleshed out. So, before we get into structures, let's talk about the cell holistically. And we left off with the statement that cells are incredibly small. And they're incredibly small for a reason. And one of the reasons that cells are so small is when they're communicating within a cell, that is, for example, when a nucleus is communicating with the rest of the cell, it does it kind of by diffusion. And diffusion is a passive process, it's not an active process at all. And so the distance between a nucleus and any disparate part of a cell becomes incredibly important. And being able to minimize that distance or increasing the speed of communication is an incredibly important thing. Additionally, when our cells are metabolically active, they're producing a whole bunch of compounds. Some of them are good compounds and some of them are bad compounds. But those compounds can either diffuse across the cell and out of the cell or into the cell, or they can be transported actively around the cell. So let's think about a cell that has, let's say, a mitochondrion that's active. Now, a cell this size is going to have to diffuse all of its metabolic products, all of the ATP that it's creating, all over the cell. And the bigger that cell becomes, right, the farther that distance it is, it's going to have to transport that cell. But there's another really important ratio that we need to keep in mind when we're talking about cells, and that's the surface to volume ratio, the surface area to volume ratio. Surface area to volume ratio is important because surface area and volume grow at different scales. Surface area, it's a two-dimensional measure, and so it grows at the exponent of 2. But volume is a three-dimensional measure, so it's going to grow much more rapidly. Now, if the volume is a measure of the amount of metabolic activity that can occur within a cell, that is, the larger the cell, the more volume it has, the more mitochondria it can have, and the more metabolically active it can be. The surface area, to some extent, is going to constrain metabolic activity because the surface area of that cell becomes the medium through which oxygen is absorbed to feed the mitochondria and carbon dioxide is released. Now, if we have carbon dioxide building up inside of our cells, that's a bad thing, and those cells can die. And so having as much surface area as possible becomes incredibly important because, again, that's like the gate across which we can get rid of all the bad stuff and we can get more good stuff in. So think about it in terms of like a city wall, right? City walls grow as a product of surface area of that wall. And if we have a ton of people inside the city's really, really large and a ton of people, the stuff that we're getting into the city, the food that's coming in through gates, is going to be constrained by the number of gates. And if we don't have enough food coming in, the people are going to starve. Now, if we have a relatively small city with a small volume and few people, then that smaller surface area will still allow enough food in to support everybody. So it's this idea of differences in growth rate. So if we were to actually graph the growth rate of volume and surface area, it would look something like this. So this is going to be surface area. I'm not gonna label our axes. Surface area grows something like this on a linear scale because of that exponent of 2. But volume grows like this, more geometrically. And there comes a point, and that point is right here, where all of a sudden the surface area is no longer going to accommodate the volume of the cell. Beyond this point, cells begin to die because they can't get rid of the bad stuff fast enough and they can't absorb the more good stuff quickly enough.
The volume doesn't
However,
accommodate the surface.
there are some ways that cells can accommodate growth and maximize this surface area to volume ratio. And we call them cell hacks. One way that cells do this is by producing long structures. That is, the whole cell assumes a really long, thin structure. And a good example of that are our neurons. So if you were to look at a neuron, it has a cell body right here that contains a nucleus. And that's most of what exists within the cell body. But then the cell extends way out like this. It's really long and skinny, and then ends in kind of like a cup sort of shape. Now that long projection maximizes surface area and minimizes volume, so that we don't run into those problems of not enough absorptive area, right? Another way that cells do that is with a structure called microvilli, and you're going to see this with intracellular structures as well. Sometimes on the surface of the cell, you can have these really long finger-like projections that stick out like this. And each of these little projections is called a microvillus. Collectively, they're called microvilli. Now what that does is it increases the surface area of that cell. And this is a really, really common structure for cells that secrete substances like digestive enzymes, or that are really, really important for absorbing substances into our body. So if you thought about it, where would you expect to see tissues that have lots of microvilli? If you know that, yeah, perfect, in your digestive system. So lining our stomach and our large intestine, we have cells that have lots of these kinds of structures because they're secreting digestive enzymes, right? That's an important part of our metabolism. And then also, you would expect to find lots of things like this within our respiratory tract, right? Because you want lots of surface area to absorb lots and lots of oxygen. Bless you.
Sorry.
No, that's just fine. These structures are incredibly delicate, and they can be damaged by all sorts of things, which is why smoking and inhaling particulates, whether it's tobacco smoke or whether it's vaping or whether it's any number of things, has such a problem with our respiratory tract because those compounds can damage these structures pretty easily. Okay. So small cells represent another problem as well, and that is it's really difficult for us to study them. And really, the problem is a problem of resolution. If you look at how the neurons and photoreceptors are structured on the back of our eyes, we have pretty good visual acuity. However, there becomes a point where 2 points become so close together that our eyes can no longer recognize them as 2 separate points. That is, those 2 points, those 2 structures, stimulate the same photoreceptor and the same neuron. That's the problem of resolution. Resolution is defined as the ability to identify two separate structures as separate structures. So as an exercise, I suppose, I could draw 2 points on the board like this. And the question is, for you at the back of the class, can you actually recognize those as two separate points? It becomes really difficult, doesn't it? But if our ability to resolve those two separate points as two separate is constrained by how our eyes work. But if we were to take that and we were to magnify it, and those points all of a sudden look like this, then we could identify them as 2 points. And so when we study cells, this process of magnification becomes really, really important. And the way that we look at cells, and we'll do this in laboratory next week, is with microscopy. So using microscopes. And there are 3 types of microscopes and microscope images that I'll show you throughout the semester. The first of which is from a compound microscope, and these are the kind that we're going to use in laboratory. They're called compound microscopes because you pass light through some substance and then through a series of lenses. And it greatly increases our capacity to resolve cell structures. In addition to light microscopy or compound microscopy, I'm also going to show you images that are a product of electron microscopy. There are 2 types of electron microscopes that I'll show you images from. The first of which is called a transmission electron microscope, and this functions very similar to the way that a light microscope does or a compound microscope does. It passes a beam of electrons through some thin slice of tissue. And the way that those electrons interact with the substance is picked up by some electron-sensitive film around the outside of the compartment that the substance is residing in. Additionally, you can have scanning electron microscopy. So with scanning electron microscopy, you're actually sending a beam of electrons at a substance, and then it's reflecting off the outside of it. It reflects off the outside of the substance because of how you prepare that substance. And what you end up doing is you end up plating that substance in gold, which is a highly reflective metal. And then the way that those electrons are being reflected allows you to generate very detailed images of some substance. So that's a scanning electron micrograph. That picture was created by a scanning electron microscope.
It looks like a spider.
It is. That is indeed what it is. Okay, so what I'm going to do is on the left side of the board, I'm going to keep a list of structures, how I would describe that structure, and then their functions. Because again, bare minimum, that's what I'm going to want you to know about each of these structures that we talk about within the cell. So the first structure is the plasma membrane. It's important to note that all cells, whether they are eukaryotic or prokaryotic, which are terms that we haven't defined yet, all cells have a plasma membrane. That is, unicellular bacteria have membranes, and all of our cells have membranes. And it serves an incredibly important function, and that is to be a selectively permeable barrier to substances. Selective permeability is important because this is what allows us to maintain homeostasis with respect to internal chemistry, right? When cells are selectively permeable, we can maintain a concentration of ions and molecules that are different inside our cells than outside our cells. It allows us to keep good stuff in, bad stuff out, and then pass bad stuff out and bring good stuff in. So the plasma membrane, its function is selective permeability. And that selective permeability, again, is important because that's what allows us to maintain homeostasis. At least one of the things that allows us to maintain homeostasis. Now, its structure, the way that we would describe it, is as a fluid mosaic. fluid mosaic. So what does that term fluid mean? Liquid, okay. And are liquids stationary or do they move around a little bit? They move around, right? So this idea of the plasma membrane being fluid means that the molecules that make up this plasma membrane are able to move around a lot. Now, that ability to move is incredibly important for cell function. As soon as those molecules become frozen in place, the cell loses its functionality and it dies. So the fluidity of that membrane is incredibly important. Now, what is a mosaic? Exactly. So it's a collection of like little pieces that could be tiles or shells that collectively make up some really, really big, cool piece of art, right? So one of the best classes I ever took as an undergrad was an art history class, and we looked at things from the Paleolithic up to almost the Renaissance. And it's the class that I did the most poorly in. I didn't do very well in it at all, but I loved it because we talked about these really, really cool pieces of art, like cave paintings in Lascaux, all the way up to really cool Grecian art. And the Greeks were really good at making these large mosaics on some of the temples that they constructed. And they were rad because if you looked at this mosaic up close, it just looked like a jumble of tiles that were thrown together. But if you got back far enough, it actually resolved into one big cohesive picture. It was awesome. Plasma membranes are also a mosaic. They don't consist of a single type of molecule. Rather, they consist of a bunch of different types of molecules. And those molecules include phospholipids and cholesterols and proteins. So, the plasma membrane is a fluid mosaic of phospholipids, cholesterol, and proteins. So we've talked about phospholipids